Welcome to the continuation of our look at cellular respiration. I know you are excited to be here. We left off last time talking about glycolysis. Let me go a step further back than that, however, because what I would like to do is remind you that cellular respiration is taking the energy that is stored inside a glucose molecule, which is a lot of energy, and essentially converting that big lump of energy into many smaller lumps, more easily usable lumps of energy that we call ATP molecules. The bulk of this is going to be happening in the mitochondria, but as we saw last time, the first hunk of it happens actually just in the cytoplasm. I want you to think of a glucose molecule as essentially a big bucket full of energy-rich electrons. And what we are doing in cellular respiration is we are breaking apart that glucose, and we are taking those energy-rich electrons, which travel with protons, so essentially what we can see moving are hydrogen atoms, but these energy-rich electrons, and shipping them somewhere where their energy will be used to help us make ATP. So that's how you need to kind of think of this. We started this out with glycolysis last time, and we learned that glycolysis is a many-stepped biochemical pathway, and we learned that it's the first of 3 pathways in this whole cell respiration process. So we just began our journey. The first third we completed. And so this podcast is going to focus on the second of these 3 biochemical pathways, which I call the Krebs cycle. It is also called the citric acid cycle. So us oldie moldies, I learned at Krebs. This is after the dude that discovered it. Citric acid cycle is simply I think this is what people call it more often now, which is more descriptive of the chemicals involved. So either way, it's the same exact thing. So let's get on with it. Glycolysis is the beginning of the stripping and breaking of glucose and stripping of its electrons to try to use their energy to get ATP. So glucose, as you already know, is a 6-carbon molecule when we draw it. During glycolysis, through a many-stepped biochemical pathway, each step catalyzed by its own particular enzyme, is broken apart ultimately into 2 chunks of pyruvate. So 2 pyruvates, each of them is a 3-carbon compound. Okay? That's essentially the end of glycolysis. We've taken glucose, we've broken it in half, and we got 2 halves, each of them with 3 carbons. And we did get a little bit of ATP out of that. We made in total 2. We actually had to spend 2, and we made 4, so our net gain was 2. We made 2 molecules of some crazy thing called NADH. All I know is that it got electrons from the glucose, and it was going somewhere. I don't know where it went. So we didn't get a whole lot of ATP, but we went through like 10 chemical steps just to break the glucose in half and get a very little bit of ATP in some molecule that we don't really know what it's going to do. That seems like a lot of work for not very much. And it is. So our job is to see now what is going to happen with this pyruvate, because there is still a lot of energy to be had in that. Pyruvate has more energy still to yield to us. If we stopped here, it would be very wasteful, because pyruvate still has a lot of stuff to give. Okay? So we need to get it out. There are still 3 carbons in each of those that we can yank electrons from. We can still oxidize those pyruvate molecules further, strip more electrons from them. So, if there is oxygen available, remember, glycolysis happened in the cytoplasm, cytosol of the cell. If oxygen is available, pyruvate will actually enter into the mitochondrion of the cell and go under further oxidation. Okay? The enzymes that make the Krebs cycle go, that further break down pyruvate, will completely break it down, completely oxidize, strip all the useful electrons that we can possibly get from this pyruvate. It will be fully oxidized by the end of the Krebs cycle or the citric acid cycle. So basically what we're going to look at now is going to take this pyruvate that we made in glycolysis, it's going to shove it in the mitochondria, and a bunch of enzymes, mitochondrion, and a bunch of enzymes are going to break it further until all we are left with is some fairly useless CO2 molecule. Useless because it has really no energy-rich electrons left to share. And so then we're going to have to see what we did with all those other energy-rich electrons that we stripped away. That's going to be the third piece of the puzzle. Okay. This is a really excellent summary diagram. It shows you really some basic but important information in a really simplified way. So here's where we started last time. We did glycolysis. We had a big old energy-rich glucose molecule. We basically succeeded in breaking it in half into 2 pyruvates. We did this in the cytoplasm. That's really important because what that tells us is that there's no fancy organelles required for glycolysis. It can just happen in the cytoplasm. All of life can do glycolysis. It is a fundamental and basic concept, okay? A process for all life that is shared. Many of the enzymes are very highly conserved among wildly different species, meaning they are very similar among wildly different species. So, we got a very little bit of ATP by means of something we learned about called substrate level phosphorylation. We stripped a phosphate off of a compound and stuck it onto an ADP and said, there you go, there's some ATP right there. We also got this crazy thing called NADH, and if you look, that's going to go to the mitochondrion as well, but this is what we're focused on right now. What happens to that pyruvate we ended up with? Well, if oxygen is present, it's going into the mitochondrion. Okay? Now, before we go, another step. We must learn about the mitochondrion. We must learn how it's structured because, remember, structure relates to how it works. It is built a certain way because it works a certain way. All right. Or maybe it works a certain way because it's built a certain way. Either way, they're related. A mitochondrion is a double membrane structure. And when I say that, what I mean is it is made of 2 phospholipid bilayers. 2 complete phospholipid bilayers. The membrane, this highly folded one that I am drawing right now, is 1 phospholipid bilayer. This is another phospholipid bilayer. That's really important, A, for how it works, but B, also, it tells us something about the origin of mitochondria. It is believed and pretty concretely genetically proven that mitochondria in fact were once free-living bacteria. Here's a little dude a long time ago, and this little guy got engulfed, yonk, by a larger cell, came up and chomped him, but instead of eating, he ended up just getting engulfed, okay, by this larger cell. And it turned out they ended up living in concert, and the mitochondrion or the bacteria did not get digested. Why? Well, perhaps this bacteria was able to provide something that this larger cell could use, like energy, and it didn't get digested. It actually got incorporated, and then this is thought to be the origin of mitochondria as organelles. This whole idea is an idea called endosymbiosis. Remember, a mutualistic symbiotic relationship is a relationship in which 2 different species engage in a relationship and both of them benefit. Endo simply means one of them is inside the other. And so Mitochondria and chloroplasts, too, are thought to have originated in this way. One was consumed by another larger cell. And further evidence of this is that mitochondria have their own DNA molecules, just like you would expect if they had once been there free-living organisms. They also can reproduce, and they do so within your cells. They reproduce, they look just like little bacteria undergoing binary fission, reproduction. So there's a whole lot of pieces of evidence that link mitochondria to this origin. You should know it. Okay, I will tell you now. Endosymbiosis, you hear that, you should think mitochondrion and chloroplasts, which we'll learn about later, right away. All right, now, sorry, having technical issues as always. All right, so this is a really sweet picture of it, all nice and computer graphic-y, but I'm going to deal with my little chicken scratch one up here. I want you to know the structure of the mitochondrion. You need to know that this part is the outer membrane, which they have nicely shown you here. This part here is the outer membrane. Okay. And this, of course, then is the inner membrane, but it's given a fancy term called cristae or cristae. I've heard it both ways. You can tell that that inner membrane is highly folded. The reason we fold anything in biology is because it is to increase surface area, and that is precisely why this inner membrane is folded. This is where the ATP ultimately will be created, and to make as much ATP as possible, we fold that inner membrane. So you can see the folds done much better here, but whatever. All right, the inner part, the little center space, if you will, if it were a donut, it's where you'd find the jelly, is called the matrix, the mitochondrial matrix, okay? And that's where we're going to be spending a lot of time for this podcast. And this is very important. The space between the inner membrane and the outer membrane, from here to here, all the way around, is called the intermembrane space. And that's going to be really important later. Not so much now, but later on. This is an actual mitochondrion, what it looks like under a transmission electron micrograph. You can see the cristae all throughout here. Okay, it's sliced through mitochondrion. Let's see. All right, so smooth outer membrane, highly folded inner membrane, cristae, intermembrane space, fluid-filled space between the membranes, got it. Matrix we talked about, inner fluid-filled space. It has its own DNA, has its own ribosome. Sorry, I didn't mention that to you, but it does. Has its own various enzymes. We're going to meet some of them. Now, this is a nice question. We talked about it a little bit with our last unit. What cells would have a lot of mitochondria? It's a good question to think about. It's a real AP-oriented question. Muscle cells, any cells that would use a lot of energy. Sperm cells, they got to use a heck of a lot of energy to try to swim where they're going. So mitochondria are found in all cells, but in particularly large amounts in high energy-consuming cells, muscle cells, and things like that. All right. This is a nice shot of mitochondria dividing, doing just like bacteria. If bacteria dividing, asexually reproducing, that's called binary fission, and that's what they're doing. So what does this tell us about the evolution of eukaryotes? Eukaryotes, cells with organelles, were likely created by one cell engulfing another. Endosymbiosis, there's the word. Please recognize it. All right. Advantage of highly folded inner membrane, more surface area. You will really see why this is so important in the next podcast. For now, we'll just kind of accept it. All right. Oxidation of pyruvate. We're going to take those two pyruvate chunks and get them into the mitochondria, because we can't do anything further with them unless we get them in there. So pyruvate then is going to have to make its way through the outer membrane, through the inner membrane to get into this region, which, if you'll remember, is called the matrix in here. Okay? Now, as it does that, it is going to pass through a protein, okay? And it is going to get oxidized as it does that, okay? And it is going to release 2 carbon dioxides as it does so. All right. I want to see if there's a better picture on here. There it is. That's the picture I want. Okay. Pyruvate. Here. 3 carbons, okay, boom, 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 and we are going to send it through a transport protein right there, a channel of, it's actually a little enzyme, and what's going to happen is we are going to strip electrons. That's NAD+, it is an electron carrier, it is going to come in and yank some electrons off, and it is going to become NADH. As that happens, we are going to release a CO2 molecule, okay, and we are going to add to pyruvate, or its remnant, something called coenzyme A. You don't really need to know what it is. You don't care what it is. I don't care what it is. Why I care about it at all is that this is where we get the name of the product that is made, and it is acetyl-CoA. All right. This is significant because this is actually what enters into the citric acid cycle, or the Krebs cycle, this compound called acetyl-CoA. So it is a 2-carbon sugar here. We're getting an NADH molecule. That's going to be important for later, not so much right now, but for later. And we also get a CO2 molecule. That is our first waste CO2 that we have made so far. When you exhale CO2, this is where it is coming from mitochondria, giving it off because of this step, and then some other steps we're going to see in a bit. So this is a little bitty in-between step. We haven't gotten to the Krebs cycle yet. This is just a transition step to get us from glycolysis to Krebs. Okay. All right. We got it. Okay. Oxidation reduction. We stripped electrons, gave them to NAD+, and we reduced that. Okay. That's the dude that figured this out. Okay, and if I had more time, or you cared more, or both, we would learn all about him. Now, this is relevant. The Krebs cycle, citric acid cycle, evolved later than glycolysis, okay, and that does make sense. Bacteria with no organelles evolved first. No oxygen in the environment. Glycolysis was the only process that could be used. We get oxygen in the environment because all the photosynthesizers evolved and put it there. Then later on, we actually get eukaryotes that can make use of that oxygen, as we'll see soon, and we get the evolution of more different biochemical pathways. So it does make sense that Krebs comes after glycolysis. But I digress. So here we are. We've got our pyruvate. It goes through its little transition step there to get into the mitochondria. And now we've got acetyl-CoA. Remember, we gave off some CO2. This is coming into the Krebs cycle. Okay? There's a 4-carbon compound that is part of the Krebs cycle that is going to come up and meet with our 2-carbon acetyl-CoA. Some enzyme's going to interact with it. We're going to get another compound. I don't really care. We're going to go a bunch of changes. There's going to be enzymes involved in making all these changes, blah, 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 blah. Okay? Remember, everything we are seeing here happens twice because we made two pyruvates. So there's two pyruvates coming into the mitochondrion, two acetyl-CoAs, and we're going to turn the Krebs cycle twice. So that is important to remember. Now, here is what is really significant about the Krebs cycle, okay? We are going to make something called citrate. I don't care. We're going to make some other things. I don't care what they are. We're going to give off CO2. That's relevant because that is where the waste CO2 comes from in the equation that we always see for cell respiration. But this is what I care about right here. At this step, one of our little NAD plus taxicabs is going to come in and strip some electrons from our carbon compounds and take them away. Where is it going? I don't care. You just need to know it's going somewhere, and it's important because what do those electrons have? They have energy. That's what they have. Okay? We're going to give off some more waste CO2. Excuse me. We're going to strip some more electrons. Boom. Stripping electrons, carrying them away. Okay? Now here, this is not completely irrelevant. It's almost is because it's so little, but we are going to make one ATP every time we turn this, and that was the point, was to make ATP, so we did get some. We've got another electron carrier that's coming in to grab electrons here called FADH2. For now, simply accept that it is very similar to NADH. It strips electrons, carries them away, but we are not going to worry about how it's different right now. And finally, we get one more go around at stripping some electrons. So we've got one more taxicab full of electrons that's going to go away. At this point, we have this 4-carbon compound created once again that will grab the next acetyl-CoA that comes in, and we spin the Krebs cycle again. What is important for you to recognize is these molecules here. NADH, we made 3 of them per turn. We made 1 ATP per turn. Remember, for every glucose, we turn this thing 2 times. So in total, per glucose, we have now from the citric acid cycle got 6 NADHs and 2 FADHs. And we have 2 ATPs. Actually, I need to put a little 2 there. So, this is a fair amount of energy. We're not sure what we're doing with it yet, because remember, the goal is to make ATP. This is not ATP. It is energy, but it's not ATP. So, how do we get those things converted to energy? Okay? All right, so we reduced them. These are all electron carriers, and we just reduced them. That means we gave them electrons. This diagram is from your book. Again, I want you to look at all these ridiculous, scary names. Citrate, isocitrate, blah, 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 blah. We don't care. We don't care. We don't care. Here's what you care about. Acetyl-CoA. You care about that there are enzymes making all this happen through every step. It's a biochemical pathway. And here is the other thing you care about. You care about that NADH and that NADH and that FADH and that NADH, and you care about that ATP. That is what you care about. Oh, wait, one other thing. The CO2s that we gave off, you care about that too. And you care that we have to do all of this twice. Okay? All right. So there's our glucose molecule. It is fully oxidized now. It has been stripped completely. We've got CO2 left, and we have ended up, because remember we do this twice, we had and we had 2 ATP from glycolysis that we made. So now we've done this twice, and we get an ATP for each turn. So now there's 2 ATP from the Krebs per glucose. So whoopee! We got 4 ATP. I got to tell you, folks, that's nothing. Okay? Glucose, 686 kilocalories per mole. Okay? And 1 ATP is 7.3 kilocalories per mole. So I got 4 times 7, 28, a little more, 29 kilocalories here out of a possible 686 that's available. That stinks. That's the pits. We need to do better. Okay? So, the point here then is what we've gotten is a whole bunch of electron carriers. They are carrying energy-rich electrons, and this is a great diagram because they have taken out all the things that you are not supposed to care about. Ooh, I forgot. Don't forget about the NADH we got in the little transition step. Okay? So, where are these energy-rich electrons going? They are going to go to the third biochemical pathway called the electron transport chain. Okay, we'll talk about what that picture is soon. So, let's see if we can account for all the energy. All right? In our glycolysis gave us pyruvate. Okay? Now, in total, remember we have the little transition step getting the pyruvate into the mitochondrion gives us one NADH, and then the Krebs cycle itself gave us 3 NADHs, so we get 4 NADHs. We get that one FADH from all of this. We're going to get some CO2 waste, 3 of those, and we are also going to get 1 ATP. Okay, now this, remember, is for 1 pyruvate. We do the whole thing 2 times because we made 2 pyruvates in glycolysis. Okay, so we've gotten a little bit of ATP. We've gotten a lot of these electron carriers. So the real value then of the citric acid cycle is not so much in the ATP that is made, but in the electrons that we have now stripped, and we are going to do something with the energy that they possess. Okay? And the place we are going to do all that is in the electron transport chain. Step on it indeed. All right, so that's the end of this podcast. So glycolysis, the point, break glucose in half, get 2 pyruvates. The Krebs cycle, the point, we get those pyruvates into the mitochondrion, we get a little bit of NADH when we do that, and we get a CO2 molecule. We create acetyl-CoA when it goes in. Then we take acetyl-CoA, push it through the citric acid or Krebs cycle, and we get 3 more NADHs, 1 FADH2, 1 ATP. We have to do everything twice, so double all of that. Now we've got a little bit of ATP and a whole lot of electrons and little taxi cabs going where? To the electron transport chain. And that's where we're going next. I hope you understand this. We'll play with it in class. Let me know how I can help. Thanks for watching. See you soon.